Good morning to all. In the series of lectures, I will be discussing about the softwares which is used for teaching pharmacology practicals and also doing research to establish the toxicity of some particular chemicals. As we know that CPC SCA is restricting the use of animals for utilization of animals for toxicity studies and also for other research purpose. So, as a part of reduction, refinement, and replacement, the utilization of the animals has to be replaced by some of the simulation techniques. So, as a part of replacement alternatives, number of softwares were available to screen the toxicity of a particular chemicals. As we know that in preclinical trials, for the determination of the toxicity, we will be going for acute, subacute, subchronic, chronic toxicity studies, and also some special term toxicity studies like carcinogenicity, mutagenicity, and reproduction toxicity. For this kind of toxicity studies, we have to utilize the number of animals. So, as a part of replacement, uh, number of softwares are available to predict the nature of the molecules. One of the software is nothing but drug likeness tool, which was originally developed by the Department of Bioinformatics of NIPER. So this drug likeness tool, it is a set of guidelines for the structural property of compounds used for fast calculation of the drug-like properties of a particular molecule. It is an open source virtual screening tool. Its calculation is based on various drug likeness to rules. For example, Lipinski rule of 5. MDDR like rules, which depends upon the number of rings, number of rigid bonds, and number of rotatable bonds. Weber rules, which depends upon the number of rotatable bonds and which determines the oral bioavailability. Goose filter, blood-brain barrier rule, and CMC50 like rules. It also includes the quantitative estimation of the drug likeness. It uses the chemistry development kit, Java library for descriptor calculations. This is very useful for the young researchers. The second software which I am going to discuss is the toxicity estimation software tool, otherwise it is called T.E.S.T, which is available as Java applications, which was originally promoted by the Environmental Protection Agency. And this is the website where you can download this particular software. And this software is used to estimate the toxicity of chemicals using QSAR methodologies. As you know that QSAR are mathematical models used to predict measures of toxicity from the physical characteristics of the structure of chemicals. Otherwise, it is called as molecular descriptors. So, this toxicity estimation software tool estimates the toxicity values and the physical properties of the organic chemicals based on the molecular structure of the organic chemicals entered by the user. For example, as you know, the nitrobenzene radical, again, the chloramphenicol is responsible for the bone marrow depression or hematopoietic toxicity. If for this, no need of any external programs. That is the peculiarity of this particular software. The users input a chemical to evaluate by drawing it an included chemical sketcher window, entering a structure of a text file or importing it from an included database of structures. So this is also a very good software for estimation of the toxicity of the prediction of the toxicity of a particular compound. The third, which I am going to discuss, is Protox 2. The Protox 2 is a prediction of toxicity of chemicals. Here you can see here the PubChem name and the canonical smiles. The canonical smiles has to be incorporated in this particular place. So by using these softwares, you can go for carcinogenicity, immunotoxicity, mutagenicity, and cytotoxicity studies, and also the nuclear receptor signaling pathways like aryl hydrocarbon receptor, androgen receptor, ligand binding domain, aromatase. All these things we can predict by using this Protox 2 software. It is mainly used for the drug design and development process. So we can easily determine the toxic doses in animals and reduce the uses of animals for the research purpose. Around 33 models for the prediction of toxicity endpoint, like acute toxicity studies, hepatotoxicity studies, cytotoxicity studies, carcinogenicity studies, mutagenicity studies, immunotoxicity studies, adverse outcome pathway. All these things we can get it from the Protox to the software. The next software which I'm going to discuss is ChemTalks software. This is another software where the toxicity can be predicted directly from the chemical structure. So by using this software, we can do Ames mutagenicity, and we can determine the LD50 values in rat following either intravenous administration or PO per oral administration and aqueous solubility directly from the structure. For this, no in vitro physical, chemical or toxicity data are required. And this can save money and time by allowing toxicity to be assessed virtually. And this also provides early stage filter for directing chemistry and prioritizing the screening methodologies. Next software which I'm going to discuss is ChemTunes or ToxGPS, and this is also based on the chemoinformatics platform. And this particular software is used to for the safety and risk assessment, and it is also it can also able to predict the mechanism of action based results and in vivo and in vitro human health and regulator for determination of the relevant toxicities. The next software which I am going to discuss is the Gastroclass. It is a pharmacokinetic model of simulation software packages which simulate IV, oral cavity, ocular inhalation, dermal, subcutaneous route, and intramuscular absorption of the particular molecules in animal population groups or in human population groups. The next software is ADMET Predictor, absorption, distribution, metabolism, elimination, and the toxicity predictor. And again, this is based on the QSAR. This is QSAR modeling software. This can able to predict the number of ADMET properties from the molecular structure. So next software is called TeleSym. TeleSym is again the quantitative system toxicology software designed to be used during drug development process to provide an indication of the potential drug-induced liver injury hazards posed by individual molecules. So with this, I have I have given an outline of some of the softwares used for the toxicity prediction. And these softwares can be useful for the young researchers doing a post graduation or PhD in the field of pharmacy or pharmacology or any basic medical sciences. Thank you. We will meet again in some other lecture.